Hello, everyone, and welcome. My name is Corinth Auld. I am Mercodia's Medical Science Liaison and the host of our webinar today. Just as a reminder, at any point, please type your questions into the Q&A box. I will hold the questions until after the presentation, at which time I will read them aloud and give our speaker a chance to answer them. So it is my pleasure to introduce our speaker, Dr. Patrik Rorsman. He is currently Professor of Diabetic Medicine at the Oxford Center for Diabetes, Endocrinology, and Metabolism at the University of Oxford. He comes to us with experience in a variety of scientific settings, as his previous positions include being on the Medical Advisory Board of the JDRF, being the Canadian Excellence in Research Chair at the Alberta Diabetes Institute at the University of Alberta, being head of the Department of Molecular and Cellular Physiology at Lund University, as well as Director and Senior Research Scientist in the Islet Research Group at Novo Nordisk. He has devoted his distinguished career in both academia and industry to the study of human pancreatic islets. His group uses sophisticated methodology to understand hormone secretion from islets at the molecular, cellular, and systemic levels. He has received numerous awards for his work, including multiple awards from the Royal Swedish Academy of Sciences and the European Association for the Study of Diabetes. He is an elected Fellow of the Royal Society and the Academy of Medical Sciences, and I might add he completed his postdoctoral work in the lab of Nobel laureate Bert Saxman. It is a privilege to have him with us today, so without further ado, Dr. Patrik Rorsman.
Okay, thank you very much for that introduction, and it's a pleasure to talk about the regulation of insulin and glucagon secretion. And in this first slide, which I hope you can see, there is a human pancreatic islet in the background, and there are some electrical signals superimposed on this image. And as will hopefully be clear during my presentation, electrical signals are very important for the regulation of insulin and glucagon secretion. The strange thing is that all the electrical signals are essential for life. They tend to talk on electrical activity that tends to send the audience off. So I will keep the actual electrophysiological recordings at a minimum, and I will try to discuss this in more general terms. But I hope to be able to convince you that electrophysiology provides us a fantastic means of understanding the regulation, the cellular control of insulin and glucagon secretion in both health and disease. Now, let's see here. And this next slide here just shows something which most of you have probably seen numerous times, which is the consensus model of glucose-induced insulin secretion in the pancreatic beta cells. And this work, of course, is mostly based on work on mouse beta cells. So the idea here is that glucose is taken up by the beta cell and metabolized in the mitochondria. That leads to an increase in the ATP-ADP ratio that in turn leads to closure of ATP-sensitive potassium channels. These channels are normally highly active in the beta cell, and they mediate an outflow of positively charged potassium ions out of the cells. So and for that reason, the inside of the cell is negative. So when the channels are closed in response to high glucose, this leads to the beta cell becoming, relatively speaking, more positive on the outside, on the inside, and that leads to, in turn, to the opening of voltage-sensitive calcium channels, and calcium enters into the beta cells, and this high calcium, in a way reminiscent of muscle contraction and neurotransmitter release, leads to the exocytosis of insulin-containing granules. And insulin secreted that way will then act on in the peripheral organs, mediate glucose uptake. There will be a decrease in the glucose concentration, and then that in turn will lead to a feedback inhibition of the beta cells. So you can see that how this machinery provides the beta cell to sense the changes in plasma glucose concentration very effectively in the beta cell. Now, I talked about the electrical activity or membrane potential of the cell. And one particular aspect of the beta cell is that it is an electrically active cell. Most people are not aware of the beta cell being electrically active, but they do, and they are. And when exposed to glucose, as illustrated here, they start generating a beautiful pattern of electrical activity consisting of bursts of action potential. That is black smear here. Now, this rhythmic electrical activity, it coincides with pulsatile stimulation of insulin secretion, as shown below in the red. Now, of course, as I alluded to, all this work was performed in rodent islets, and as my clinical friends and colleagues here in Oxford never tire of reminding me, rodent diabetes is not a major clinical problem, and if there are mice in the hospitals, it's a pest control problem and not a medical problem. So we decided to do something about this a few years ago, 10 years ago, and we were very fortunate because here in Oxford, it's one of the 2 or 3 national islet transplantation, clinical islet isolation and transplantation centers. Of course, these people, these colleagues, they don't isolate human pancreatic islets for research purposes, but there is a need for quality assessment, and that gives us the opportunity to characterize human islet cells. I emphasize that we can only do this if there is research consent, and I also emphasize that we don't need that many islets, so we don't really compromise the transplantation work. So we are actually content with a few islets that stick to the walls of the test tube. But since the start of this of the islet transplantation unit in 2006, we have analyzed more than 230 normal human pancreases or human islet preparations, and we had a few, as shown below here, a few islet preps from diabetic donors, 12 from type 2 and 1 from type 1 diabetes. So, and we of course worked on this, and we have mainly in my lab used electrophysiological techniques and secretion measurements, and this slide here is just to emphasize really that mouse and human beta cells are not the same, and you don't really have to look into the details of this, but you can see that there's a lot of red in this slide, and the red indicates important differences between human and mouse beta cells. And looking at this, I mean, we have to confess that we despaired, and we almost had to conclude that all the work that we had spent on working or trying to elucidate the functions of rodent beta cells was almost wasted, I mean, in terms of understanding the behavior and function of human pancreatic islet cells. And because of these important differences, I think most of the data we present today will be based on our work on human pancreatic islets. Having said that, I emphasize that mouse work is not useless. It's just that we have to start with observations in man and then turn to our furry friends to address very specific questions. But we should not start in the mouse and expect everything to be the same in mouse. So the differences between human and mouse islets we can see at every level. It starts even at the level of the architecture of the pancreatic islets. As many of you probably know, mouse islets have a very well-organized architecture with a central beta cell core and then surrounded by a cortex of non-beta cells. But in human islets, as illustrated here, the picture is very different. It's more chaotic, and the beta, alpha, and delta cells here, color-coded, they are more mixed. And this, of course, facilitates cell-to-cell communication and also paracrine signaling within the pancreatic islets. And we can, of course, also take these islets and measure hormone release. And as you can see here, to the left, we have the responses in mouse islets, and you can see that where we increase the glucose concentration, there is a stimulation of insulin secretion when we go beyond 5 millimolar. But in human islets, shown to the right, we see stimulation of insulin secretion already at 3 millimolar, and then it increases rather steeply at higher glucose concentrations. the vertical lines here indicate the normal plasma glucose concentrations. And you can see that at these normal fasting plasma glucose concentration, there is already significant stimulation of insulin secretion, both in man and in the mouse. Now, the interesting thing is that if you take out human islets and transplant them into a diabetic mouse, they will control the plasma glucose concentration to human levels. So rather than having 8 millimolar, as is normal for a mouse, the glucose concentration in a mouse that has been transplanted with human islets will be around 4 millimolar. So that illustrates that it's the properties of the pancreatic islets that determine the plasma glucose concentration rather than perhaps the more systemic effects. So the performance of the pancreatic islets is essential. Well, glucose stimulates insulin secretion, and the human pancreatic islet and the human beta cell is, like its mouse counterpart, also electrically active. There have been very few recordings from human pancreatic islets, and here we have one of the few successful recordings that we managed to obtain from an intact human pancreatic islet using a recording electrode shown up to in the right corner. As you can see here, that when we increase the glucose concentration from 1 to 6 to 8, 11, 16, and 22 millimolar, there is stimulation of beta cell electrical activity, and you can also and it's evident that even a glucose concentration as low as 6 millimolar glucose evokes very strong electrical activity. And the part of the recording contained here by the red rectangle is shown on an expanded time base below. And what is interesting here is that that there are some time-dependent changes in electrical activity. The first period of electrical activity it shows these bursts or oscillations in membrane potentials, and they also have large amplitude. Then after a while, you go into a period of continuous action potential firing, but of lower amplitude, and finally, electrical activity resumes. Oscillatory electrical activity resumes, but with a slightly higher amplitude than before, but lower than the initial responses. And so that is what I've shown here, and I will come back to the possible significance of that later. Just pointed out already now. So based on all this work that I've shown you, we have come up with a revised consensus model of the stimulus secretion coupling in the human pancreatic beta cells. And many of the things here look very familiar, very similar to what you see in the mouse beta cells. There are the KATP-channels, there is glucose metabolism, there are the voltage-gated channels, and there is, of course, insulin secretion. But there are very notable and important differences, and they are highlighted by the in red here. So for starters, the glucose uptake is not mediated by GLUT2 as in the mouse beta cell. It's mediated by GLUT1, which might be significant because GLUT1 operates at lower glucose concentrations than GLUT2. The second major difference is that whereas the mouse beta cells have a high resting activity of the KATP channel, here quoted as conductance, which I'll explain later, it's less than 10% what we see in the mouse in human beta cells, and that is, I think, very important. The types of voltage-gated ion channels in the human beta cells is quite different from what we see in the mouse beta cells. So there are other types of calcium channels and sodium channels and potassium channels than the ones that are active in the beta cells. And of course, but there are also things which are the same. And so the calcium goes up into the cell, inside the cell, and that triggers insulin secretion the same way as it happens in the beta cell. And of course, you can ask the question: does it matter whether it's the a PQ type calcium channel as in the human cell or an L type as in the mouse? And of course, and it probably doesn't matter much to the human beta cell, but it matters to us if we are trying to understand what how insulin secretion is regulated in the human beta cell and how things might go wrong in the human beta cell. Then we have to know exactly which molecular subtypes of the ion channels which are important for secretion. So when all these things have happened, when we have stimulated the beta cells with high glucose, then we start generating action potential firing in the human beta cells, and they can be, as shown up to the right here, they can be quite beautiful, very neuronal-like, and we also get biphasic insulin secretion in these cells. Now, the interesting thing about this model is, I think, that from these differences which are highlighted in this slide in red here, we can actually explain why human beta cells start secreting insulin at a lower glucose concentration. But before doing that, let me just briefly point out also, which I forgot to do, that the KATP channel is, of course, also the target of the hypoglycemic sulphonylureas, and these drugs, they block the KATP channels by a direct interaction with the channel protein, which is independent of any effects on glucose metabolism. Now, let's go back to the question I alluded to before. Why is the threshold for insulin secretion in human beta cells lower than that in mouse? And we think that this is because of the differences I highlighted in the previous slide. There is, to begin with, less activity of the KATP channel, and that means that the human beta cell is partially depolarized already in the absence or at low glucose compared to the mouse beta cells. And then in addition, they have a subtype of calcium channels which open at more negative voltages than membrane potentials than the counterparts in mouse beta cells. And they also have large voltage-gated sodium channels that also provide a depolarizing force. And these things together, they make it easier for the human beta cell to start generating action potentials and therefore trigger insulin secretion at low glucose concentration than it was possible in mouse beta cells. Well, when all these steps have happened, when glucose has been metabolized and we generated electrical activity and calcium has gone up inside the cell, then of course you get insulin secretion, and insulin secretion, as we all know, is biphasic. And to understand what causes this biphasic nature of insulin secretion is, of course, very important. And I'll come back. There's a physiological significance to this, as I will discuss later as well. One idea that we've had is that the biphasic nature of insulin secretion reflects the existence of different pools of secretory granules. There are about 10,000 secretory granules in the beta cells, but only a fraction of these are immediately available for release. And we call these the readily releasable pool of granules. And the jury is out at the moment on whether these readily releasable granules are situated just below the plasma membrane, or if they are docked, or whether they are situated somewhere some distance away. But I think most people probably agree that they must be situated in reasonable proximity of the plasma membrane. And what we believe is that it's these green readily releasable granules that undergo release first in response to calcium influx through the voltage-gated calcium channels. And when this readily releasable pool has been depleted, then new granules have to be mobilized. And so you have to convert these red granules into green granules. And that is a process with what we call mobilization. It could be just a chemical modification of granules which are already situated below the membrane, but it could also involve a physical translocation from regions within the cell. In both cases, this process of mobilization is dependent on ATP, produced in response, for example, to glucose stimulation, or cyclic AMP, which is, of course, a product of GLP-1 stimulation. And when both these factors are present, then granules, the reserved granules, become available for release and undergo release. And this mobilization then is is rate limiting to secretion, and that will be the reason why second phase release is slower than the peak rate observed during first phase insulin secretion. So we can look at this and test the effects of different stimuli on insulin secretion using a perfused pancreas preparation. And first we used here glabenclamide, which is a sulphonylurea, and that acts directly on the KATP-channels. And when we apply glabenclamide at a low glucose concentration, with just 1 millimolar, you get a very sharp stimulation of insulin secretion. But the response is almost entirely transient, and within 5 or 6 minutes, insulin secretion has returned almost to baseline values. It's just slightly above what you see under control conditions, as indicated here by the red horizontal line. While the different results are obtained when we instead stimulate the islets with high glucose. And that is the red phrase here. First of all, as you see here, the response is delayed, and that delay, of course, reflects the time it takes for glucose to be metabolized within the cell. And then you generate or produce the first phase insulin secretion, which looks very similar to the one that you obtain with glabenclamide, but it's slightly longer. And then what is really obvious is that when you stimulate the cells with glucose, you have a very prominent second phase insulin secretion. And this large or sustained second phase insulin secretion we attribute to the continuous mobilization of new granules from within the cell by ATP generated during the metabolism of glucose. It's very interesting to compare these rates of test this concept and looking at insulin secretion from in vivo in human subjects. And here I've taken data, published data by a paper published by Kashyap et al. In diabetes 12 years ago. So here they measure the insulin secretion rate in response to a glucose elevation from a basic glucose, which is 4.5, up to about 12 millimolar, if I got the numbers right. And as you can see, in these human islets in vivo, they get a very nice biphasic response of insulin secretion. And using our conservative values for the number of islets per pancreas, number of beta cells per islets, and knowing that each secretory granule contains about 1.6 at the most of insulin, we can actually convert the insulin secretion rate to picomoles or granules per minute. And doing that, we can estimate that the basal rate of secretion at 4.5 millimolar is about 0.2 granules per minute. The peak rate at 12 millimolar glucose during first phase secretion is about 4 granules per minute, and second phase insulin secretion is about 1 granule per minute. And based on this, and then you can compare this with the ultrastructure of the pancreatic beta cell. This is an EM of a human pancreatic beta cell. These structures here, they are the secretory granules. The nucleus is up here, and these black dots here, that's the insulin crystals. A human beta cell contains about 10,000 secretory granules, and 700 of these granules are situated within 200 to 300 nanometers from the plasma membrane, which is just indicated here by the red line. So now taking these numbers and then doing some trying to estimate how many granules of insulin, how many insulin granules you need per day. That actually, and assuming that we eat 3 times a day and 3 main meals and 1 snack, we can estimate that the total release of insulin granules released per day is about 550 granules per beta cell. And that is, of course, as you might remember, less than the granules which are situated within 1 granule diameter from the plasma membrane, which is about 700. So the granules which you wake up with in the morning that are situated within at the plasma membrane, they are almost enough, or they are enough, to take you through the entire day. And the total daily requirement of insulin corresponds to about 5% of the granule content. It's not that much different between beta cells from non-diabetic people or diabetic patients. So it's actually quite difficult looking at the ultrastructural picture of the beta to understand how you can get diabetes at all. So there is sufficient insulin in the beta cells to cure diabetes many times. But what I think one important message that we can get from this is that the reported 30 to 40% reduction of beta cell mass that is unlikely to have much effect or much negative effect on insulin supply. There will still be plenty or more than enough of insulin within the remaining beta cells to cater for the insulin needs of the body. So why is insulin secretion biphasic? This is, of course, a very important question. And the reason that this is important is, as I will discuss later, is that biphasic insulin secretion is lost in type 2 diabetes. And the loss of first phase insulin secretion is actually one of the earliest signs of type 2 diabetes. Biphasic insulin secretion is something which we can trigger with glucose, but it's nothing unique to glucose. And basically, any stimulus will evoke biphasic insulin secretion, albeit to slightly variable extent. Glibenclamide, for example, as I showed you in previous slide, the response to glibenclamide almost entirely consisted of first phase insulin secretion with very little second phase insulin secretion. But what is striking is that in type 2 diabetes, there is a loss of glucose-induced first phase insulin secretion. So the idea here, or the justification for trying to understand what causes biphasic insulin secretion, is that will give us some clues as to what might go wrong in type 2 diabetes. So I will devote a few minutes trying to explain why this is. So you remember that a few slides ago, this is the same electrical recording that I showed you a few slides ago, that I pointed out that when we stimulate electrical activity with glucose, you have these time-dependent changes in the amplitude of the action potentials. The first action potentials were quite big, and then you had this period of very smaller action potentials, and then finally action potential finally resumed that was slightly bigger than before. And what we wanted to find out is how does this correlate with the efficacy at eliciting exocytosis. And one way that we can do this is to use a biophysical technique of measuring secretion. So rather than measuring insulin secretion biochemically, which is of course a traditional and gold standard approach, we can use a biophysical technique to measure insulin granule exocytosis, which is based on the fact that there is a conversion factor, that the biophysical property capacitance of the cell is proportionally related to the cell surface area. So when granules fuse with the plasma membrane in response, for example, to an action potential or a voltage depolarization, there is an increase in the cell surface area that we will detect as an increase in capacitance. And in the lower left corner here, we have now plotted the responses that we obtain when we stimulate the beta cell by voltage pulses going to different member potentials. And as you can see, there is a very steep relationship between the amplitude of the and the exocytotic response. So the maximum we observe at around 0 millivolts, which is very close to where the beta cell action potential normally takes the beta cell member potential, but then as we go more negative, there is a very dramatic decrease in the amplitude. And actually, below 0 millivolts, there is a 5% decrease in exocytosis for every millivolt that we reduce the amplitude. So now we can take the electrophysiological response we have up here and try to predict how these action potentials, how good these action potentials will be at eliciting exocytosis. So we take the responses here at 1, 2, and 3, and here down here I plotted the expected exocytotic responses. And as you can see, the first action potentials are very good at eliciting exocytosis. These action potentials over here, although they are generated at high frequency are almost useless at eliciting action potentials, and these intermediate ones, they are somewhere at the middle of the curve. So if you plot these action potentials at the same time course as the response up here, you get a reasonable biphasic response of insulin secretion. And I think that this has big implications for the understanding of type 2 diabetes. So let's now consider what causes type 2 diabetes and what is type 2 diabetes at the level of insulin secretion. So when we measure insulin secretion from the 12 diabetic preps that we have had the opportunities to study, we found that insulin secretion in these diabetic eyes, it's much reduced compared to what we see in the non-diabetic donors. But the important thing to notice is that although the response is reduced, they are not completely refractory to glucose. They still respond, but the response is too small. And if you plot this, the stimulation of by glucose, and you quote it as a stimulation index, which is basically glucose insulin secretion at 20 glucose divided by that at 1 glucose, you can see that the stimulation index is reduced by about 60%. And now, important thing here to notice is that I've quoted or expressed the insulin secretion here as percent of content. So that means that this reduction of glucose-induced insulin secretion cannot be explained by a reduction in insulin content. It must be due to an impaired function of the beta cell. And a big question is, of course, which function of the beta cell is impaired. But before I do that, I will also spend a few words on this eternal question whether diabetes is caused by or involves a reduction of the beta cell numbers. Because this is frequently stated in the literature and at meetings. And I think that the problem is that you can't really decide it. Although there are reports that in that type 2 diabetic patients have maybe a 30 to 40% reduction of their beta cell numbers. We don't know whether they got diabetes because there was a destruction or a loss of beta cells, or whether they had an increased risk of developing diabetes simply because they were born with fewer beta cells to begin with. And I call this the Usain Bolt effect of diabetes. Because, I mean, although I have no scientific evidence, and it's very unscientific, all this, I suspect that one of the reasons that Usain Bolt is so successful running is that he has maybe long legs, or at least if he had very short legs, he would have been much less successful winning gold medals. And the same thing, I think, it is with the beta cells. If you are born with many beta cells, you will almost certainly win the diabetes race, because the risk of getting type 2 diabetes is much reduced. So coming back now to what is the mechanism, what happens in type 2 diabetes? Well, this is, I already mentioned that in type 2 diabetes, the beta cells are not completely refractory to glucose. They still stimulate, glucose still stimulates insulin secretion, but what happens is that you have an almost complete loss of first phase insulin secretion, and second phase insulin secretion is significantly reduced. Why does this happen? And of course, in order to do this, we would very much like to do electrophysiological recordings from diabetic beta cells. But as you would probably understand, these experiments are not that easy to perform, because it's very difficult to obtain diabetic human beta cells or islets. And the main reason for that being that they are, of course, not used for transplantation purposes. They would have to be isolated for research purposes only. But here is one of the few recordings that we have here, down in red. In the top part of it, I show you the same recording I showed you before. It was taken from a normal pancreatic islet, and below here we have the same response from a diabetic islet. And what is very striking here with these diabetic beta cells is that they generate spontaneous action potentials already in the absence, or at very low glucose concentrations. What is also evident is that when we stimulate with 6 millimolar glucose, which in non-diabetic beta cells elicited a very strong stimulation of electrical activity, there was only a small stimulation of electrical activity. And even when we go to 10 or 20 millimolar, which has a very strong effect in non-diabetic beta cells, although there is some stimulation, it's not nearly as strong as the one that we get in the non-diabetic islets. And another thing is that I want to point out is that in the type non-diabetic ones, there is of course also this reduction in the amplitude of the action potential high, which is important for the generation of the reduction of the fact that second phase insulin secretion proceeds at a lower rate. We don't see that so much in the diabetic beta cells. What is the reason then for this impairment of glucose-induced insulin secretion? So here, one possibility would be that there is not enough ATP being generated in the diabetic beta cells, and there are actually data supporting this idea. This is taken from the paper published by Guerra et al. In Diabetes 10 years ago. So here is the increases in the ATP-ADP ratio in non-diabetic human islets, and as you can see, increase in the glucose concentration has a good stimulatory effect on ATP-ADP ratio in the non-diabetic islets. But in the type 2 diabetic islets, there is no detectable stimulation. And what is also notable is that the basal ATP-ADP levels are higher in the type 2 diabetic islets than in the non-diabetic islets. And we speculate that this high basal ATP-ADP ratio might explain the occasional firing of action potentials at low glucose concentrations in diabetic islets. So this will then try to schematically explain the differences between the normal non-diabetic beta cell. This is the same thing as I showed you before. This is what happens when we start at low glucose, KATP-channel activity is high, the beta cell membrane potential is negative, and therefore there's no electrical activity, and insulin secretion for that reason is kept low. When we increase the glucose concentration, ATP is being generated inside the cell. It closes the KATP channels, as seen schematically over here. This leads to membrane depolarization and the initiation of electrical activity. The action potentials resulting from this, they are initially high, but after a while, the amplitude is reduced. And this correlates with and let's explain this biphasic response. The first phase insulin secretion is due to the firing of the larger amplitude action potentials. Second phase insulin secretion is due to the firing of action potentials of slightly lower amplitude. Now, what happens in the diabetic cells? Well, we say that we speculate, and that's based on the evidence published by Guerra, is that there is an impaired generation of ATP in the beta cells. But it already stopped at a slightly elevated level. So for that reason, the KATP channel activity is slightly reduced. So the dotted line here is the non-diabetic, and the continuous line is the one in type 2 diabetes. And you can see that. So the KATP channel activity, because of the slightly higher basal ATP-ADP ratio, is lower than in the non-diabetic cells, and therefore the diabetic beta cell is slightly depolarized, generates the occasional action potential, and basal insulin secretion, because of that occasional firing of action potential, is slightly elevated compared to the non-diabetic controls. When we increase the glucose concentration in the diabetic beta cells, there is not that much of an increase in the ATP-ADP ratio, so the reduction of the KATP channel activity is very small, and for that reason there is not much stimulation of electrical activity, and for the same reason there is not much stimulation of insulin secretion. We lack this initial bout of intense electrical activity, and that explains the loss of first phase insulin secretion. What happens now if we try to restore normal KATP channel activity in the beta cell. And that is, of course, what sulphonylureas do. And so this is schematic what happens in the diabetic cell when we apply a sulphonylurea, which closes the KATP channels and triggers electrical activity. First bout of intense action potential firing of high amplitude, and then after a while, the amplitude of the action potentials is reduced, and that explains this loss. And you have the second phase of insulin secretion proceeding at a lower rate than the peak rate observed during the initial phase of secretion of electrical activity. And that, of course, then explains why sulphonylureas restore biphasic insulin secretion also in people when you make the same experiments in diabetic people. What you can observe here also is that the amplitude of second phase insulin secretion is lower even in the presence of sulphonylureas, and we explain that we attribute that to an impaired mobilization of secretory granules in the beta cell. So there is impaired mobilization of granules from the reserve pool into the readily releasable pool. And maybe the impaired generation of ATP in diabetic beta cells would also explain that. I think I have to, in the interest of time, I have to skip the next slide, which was about GLP-1, and then I move on to the glucagon aspect, which I find very exciting and very important, and because diabetes is not just a matter of impaired insulin secretion. To the right here, that this is just the old data I showed you in the previous slide, showing the impaired insulin secretion, reduction of glucose-induced insulin secretion. To the left here, I show the glucagon secretion measured in the same samples. And you can see that when we increase the glucose concentration from 1 to 20 millimolar in non-diabetic islets, we get the expected inhibition of glucagon secretion, which is, of course, exactly what we expect and want, because glucagon is a hyperglycemic hormone. It's only needed at low glucose concentrations. But when we repeat the experiments in type 2 diabetic islets, we don't see this inhibition. There are a few things we can. What we see is instead a stimulation of glucagon secretion, and in other words, I mean, there are actually two defects of glucagon secretion. There is first the reduction of insulin secretion at low glucose, and there's also this inverted response to high glucose, which I just mentioned. These things, they exactly echo the disturbances that we see in vivo in clinical studies. And this reduction of glucagon secretion at low glucose is actually very important. This explains the impaired counter-regulation, or it might explain the impaired counter-regulation that bedevils, I think, insulin therapy in many diabetic people, and that is a major cause of hyperglycemia, a condition that is potentially fatal, and is actually one of the major causes of mortality in insulin-treated diabetic people. And the reason for that is that glucagon secretion, or it appears to be that glucagon secretion in diabetic islets is much lower under hyperglycemic conditions compared to what it should be and what we see in non-diabetic islets. So in order to understand what these abnormalities, or this dysregulation of glucagon secretion, we argue that you have to understand the normal regulation of glucagon secretion. So what do we know about the normal regulation of glucagon secretion? Well, this is a hugely controversial area. If there is reasonable consensus about the regulation of insulin secretion, there is an almost complete lack of consensus when it comes to the regulation of glucagon secretion. But one very popular idea is that the alpha cell, here in blue, is under paracrine control of either of the beta cell or the delta cell. So the idea here would be that glucose stimulates insulin and somatostatin secretion, and these hormones then act on the alpha cell in a paracrine fashion to suppress glucagon secretion. And the alpha cell would not have any intrinsic regulation on its own. So we wanted to study this, and if this hypothesis is correct, and it has many very distinguished proponents, then there should be a direct correlation between the stimulation of insulin secretion and the suppression of glucagon secretion. So we tested this using a perfused mouse pancreas preparation, where we perfused the mouse pancreas in the normal direction and at a physiological rate. So it's as close as you can get to the in vivo situation, but was working ex vivo. And what we did here was that we increased the glucose concentration from 1 to 6 and 20 millimolar. We measured glucagon in blue and insulin in red, and plotted it against time. And as you can see, that when we increase the glucose concentration from 1 to 6 millimolar, glucagon secretion is reduced from a high level down, and from a much lower level. It's reduced by about 70%. And at the same time, insulin secretion is stimulated with a delay, but the increase is fairly small, and then it settles at a much lower level than the peak value during the second phase release. But there is no change in glucagon secretion when insulin secretion is reduced during second phase insulin secretion. When we increase glucose to 20 millimolar, there is, of course, a much stronger stimulation of insulin secretion, but as you can see here, there is no further inhibition or very little further inhibition of glucagon secretion. And finally, when we remove glucose, there is, of course, a reduction of insulin secretion and a recovery or return of glucose secretion to the high levels that we have previously observed at low glucose. Now, if you look at these records very carefully, you can see that there's an interesting dissociation between glucagon secretion and insulin secretion just after increasing the glucose. So the alpha cells or glucagon secretion is very promptly reduced in response to high glucose, and the inhibition is almost complete before there is any detectable stimulation of insulin secretion. So for these reasons, we think that the alpha cells must possess some sort of intrinsic regulation, and they do not strictly depend on paracrine inputs from the beta cells. And you can make a similar argument for a lack of effect of the delta cells. Most of the inhibition of glucose secretion occurs before there is any detectable stimulation of somatostatin secretion, and indeed you can antagonize somatostatin, the action of somatostatin, using somatostatin antagonists or antibodies, and still glucose inhibits glucagon secretion. So altogether, these recordings or measurements suggest that the beta cells or the alpha cells must possess an intrinsic regulation of intrinsic glucose regulation of glucagon secretion, and that the paracrine signaling is perhaps not quite as important as we formerly believed. But I hasten to add that the idea that the alpha cells possess an intrinsic regulation does by no means exclude the contribution, or does not exclude that they also are subject to a paracrine regulation. I think that is, by contrast, or I think that the paracrine regulation might actually be quite important under certain conditions. But the aspect I want to cover here is the nature of the intrinsic regulation. Now, as you can see here, the alpha cells, like beta cells, are electrically active, which is, of course, good news for us, because we are electrophysiologists and we like to work with electrodes. But they are different from beta cells in that they generate action potentials already at low glucose concentrations. At 1 millimolar glucose, you would not see any electrical activity in beta cells, but as you can see here, the alpha cells, they generate very nice action potentials at low glucose. These action potentials, they reflect the activity or activation of several types of voltage-gated channels. You don't have to pay any attention to this. The only channel which is important and that you might wish to remember is the voltage-gated sodium channel. And the reason for that will hopefully become evident. The voltage-gated sodium channel is a very important channel. It's in nervous conduction and in impulse propagation in the heart. And one of its main characteristics is that it has three different states of activity. That it's, of course, most of the time it's closed, not active, but then it can open in response to a change in the membrane potential, and then of course this is where it mediates sodium influx into the cell. Now, if you maintain this stimulation or the depolarization, then sooner or later, usually sooner, it goes into a different closed state, which is called the inactivated state. And this state is functionally distinct from the normal closed state, but of course it's similar to the normal closed state in that it mediates no sodium influx and it will have no impact on the membrane potential. And in order to become active again, the sodium channel has to be reactivated, and this reactivation that requires a strong repolarization. So the channel has to see a negative membrane potential again in order to return to the closed state from which it can open again. This reaction is irreversible, and this is, I think, it turns out to be quite important. And I will come back to that in a few minutes. Now, this is, again, just to remind you of the stimulus secretion coupling of the beta cell. And I don't have to remind you now about how the beta cell recognizes glucose, but the beta cell is the tip of the glucose sensing cell. And it's, I think, reasonable to assume that there will be similarities between the glucose sensing of the beta cell and the alpha cell. And indeed, when we start looking at the alpha cell, it's striking how many things are similar between the beta and the alpha cell. And one of the paradoxes is that the alpha cell has a very high density of ATP-sensitive potassium channels. And we know that in beta cells, closure of these channels are essential for the ability of the beta cells to for glucose to stimulate insulin secretion. But what are they doing in the alpha cells, and how could they contribute to the inhibition of glucagon secretion in alpha cells? So we tested that using a sulphonylurea. In this case, it's tolbutamide, which is good to use in the test tube. We used again the diffuse pancreas. We measured insulin glucagon secretion at 6 millimolar, 1 millimolar, and following the addition of tolbutamide. So at 6 millimolar glucagon secretion is of course already partially suppressed. When we lower the glucose concentration to 1 millimolar, we get stimulation of glucagon secretion. That's an approximate doubling of glucagon secretion. And finally, when we apply tolbutamide, we get suppression of glucagon secretion again. So what this suggests is that KATP channel activity is somehow required for glucagon secretion to occur, and that increased KATP channel activity somehow stimulates glucagon secretion. So it's the opposite to what we observe in the beta cell, where inhibition of glucagon secretion of the KATP channels leads to stimulation of insulin secretion. So how can we explain this? So we resorted to our favorite technique. We measured electrical activity in alpha cells, and this is the sort of recording we get. So at 1 millimolar glucose, the alpha cells are very, very active, and this, of course, correlates with high rate of glucagon secretion. And then we applied tolbutamide, which I remind you led to inhibition of glucagon secretion when we measured it in the diffuse pancreas. But paradoxically, what tolbutamide does on electrical activity is not to inhibit glucagon or electrical activity. It appears, if anything, to stimulate action potential firing in the alpha cells. You can see that there's more black stuff here than over here. Now, if we look very carefully at these action potentials, you can see that the action potentials recorded at 1 millimolar glucose alone, they are much bigger than the action potential that we see at in the presence of tolbutamide. And interestingly, we see the same sort of reduction if we apply a compound called TTX, tetrodotoxin, which is a blocker of voltage-gated sodium channels. And TTX, it has like glucose and tolbutamide, it also inhibits glucagon secretion. So what this suggests is that glucagon secretion in the alpha cells is very much dependent on the activity of voltage-gated sodium channels, and these are channels that inactivate. So what we are postulating is that these channels inactivate in response to depolarization, and that explains why the action potential amplitude is reduced. And the reduction in amplitude of the action potential then explains the reduction of secretion. Why is that? Well, by now you are hopefully such a distinguished biophysicist that you're already acquainted with the capacitance measurement technique. So what we have done here now is that we have measured exocytosis, glucagon exocytosis in alpha cells in intact mouse pancreatic islets, and we have measured it using stimuli which simulate action potentials firing at low glucose and at high glucose or in the presence of tolbutamide. And you can see that the large amplitude action potentials, they are very good at eliciting glucagon granule exocytosis, whereas the small action potentials, they are much less effective at eliciting glucagon granule exocytosis. And in fact, this reduction of action potential high that we observe in the presence of glucose, high glucose, or tolbutamide, they lead to a 75% reduction of glucagon exocytosis. We have measured KATP-channel activity over a wider range so altogether these data suggest that KATP-channel activity is very important for the regulation of glucagon secretion. So we then try to determine exactly what does glucose and tolbutamide do on KATP-channel activity in alpha cells. And the problem is, and it's even debated whether KATP channels are at all active in the in alpha cells. So we made measurements of KATP channel activity in alpha cells in metabolically intact islets, or in the alpha cells in intact pancreatic islets, and using the perforated patch whole cell technique. This is just technical detail. It's not so important. But the important thing is that we can combine this with membrane potential recordings of electrical activity and also measurements of glucagon secretion. So the next slide here just then summarizes what we observed with high glucose or with tolbutamide. And here we have expressed the data or the channel KATP channel activity as conductance. But the only thing you have to remember or understand is that a high conductance corresponds to a high activity of the KATP channel. So when the conductance goes down, that corresponds to a decrease in channel activity. And indeed, when we stimulate or when we expose the alpha cells to high glucose or to tolbutamide, we see a decrease in KATP channel activity. And we think that this reduction that we observe in response to glucose does indeed reflect closure of KATP channels, because when we apply tolbutamide first and then glucose, there is no further reduction of KATP channel activity. And tolbutamide being, of course, a blocker of the KATP channels. But the effects are extremely small. And the effects are limited, the glucose effects are limited to 50 picosiemens. To most people, this doesn't mean much, but that corresponds to the activity of 4 channels in the entire cell. And it's much, much lower than the corresponding changes that you observe in beta cells, which are 100 times bigger. So that overall that suggests that the KATP channels in alpha cells are under very strong tonic inhibition. So now we can try to explain the differences between the alpha cells and the beta cells. And what will be evident here is that the beta cells and alpha cells are actually very similar. So to the left here we have the beta cells, and to the right we have the alpha cells. And in the beta cells we have the high KATP channel activity at low glucose, low negative membrane potential, no electrical activity, low secretion. And then when we increase glucose, KATP channel activity goes down, electrical activity is stimulated, and you get stimulation, biphasic stimulation of insulin secretion. When we repeat experiments in the alpha cells, we measure a very, very low activity of the KATP channels. And that, of course, explains in turn why the alpha cell, or how the alpha cell is capable of generating spontaneous action potentials already in the absence of, or at low glucose concentration. And because of the high rate of action potential firing, the alpha cells are secreting glucagon at a very high rate. What is interesting, what happens now when we increase the glucose, is that we produce further inhibition of KATP channel activity. So it goes from almost nothing to nothing. And that, as in beta cells, this leads to a membrane depreciation, similar to what we have showed you before we took glutamine. This leads to stimulation of electrical activity, but it also, very important, it leads to a reduction of action potential height. And as I showed you before, and the reason for that is that the action potentials are important because they trigger the opening of calcium channels, which are very tightly linked to glucagon exocytosis. So when you have a reduction in the amplitude of the action potentials, you have much less activation of these channels, and that is the reason you have this steep dependence or relationship between membrane potential, the amplitude of the electrical stimulus, and glucagon granule exocytosis, as I showed you in the previous slide. And the net result of that is that you get a suppression of glucagon exocytosis. You might notice here that the first thing that you see when you apply high glucose and you have this transient stimulation of electrical activity is that you actually get a slight transient stimulation of glucagon secretion, which is actually what people actually have observed in the perfusion measurement. And if you go back to the perfusion experiments I showed you, there was actually an indication of a transient stimulation of glucagon secretion upon increasing the glucose concentration. But then eventually, when the action potential amplitude has been reduced, there is a very strong suppression of glucagon secretion. And the increase in action potential frequency is not sufficient to compensate for the strong inhibitory effect due to the reduction of exocytosis due to the reduction of the action potential height. So the important thing here is that the important difference between the beta cell and the alpha cell is really that the KATP channel activity in the alpha cell is much, much lower in the alpha cell. And that explains everything. But apart from that, the beta cells and alpha cells are remarkably similar. And actually, I mean, the decrease or the transition between first and second phase secretion in the beta cell is very similar to this suppression of glucagon secretion. And the mechanism is the same. It's due to a reduction in action potential height. So beta cell, so we can say that in other words that the alpha cell glucagon secretion is amplitude modulated, not frequency modulated. In the beta cell, it seems to be a little bit more about frequency modulation. But the reason that we still have insulin secretion under these conditions is simply because you start from a very, very low level to begin with. There's hardly any insulin secretion to begin with. So even these action potentials, which are not very good at eliciting exercise, they will still lead to a stimulation of insulin secretion above basal. Not so in the alpha cells. Here you go below the basal glucagon secretion. So I know I understand. I'm painfully aware that I'm talking too much, but we then used another trick that to measure or the relationship between the KATP channel activity and glucagon secretion over a wider range of KATP channel activities. And the way we did that was to use a pharmacological agent called diazoxide, which is an activator of the KATP channels. So just now to familiarize you with this plot here, is that we plot here KATP channel activity, again expressed as conductance. And we have started the experiment at 1 millimolar glucose. That, of course, has a rather slightly elevated KATP channel activity around 300 picosiemens, as you can see here. Then we increase the glucose concentration to 6 millimolar, and as you can see, this leads to inhibition of glucagon secretion from here down to here, and it also reduces the KATP channel activity. What we do then is that we, in the continued presence of glucose, apply increasing concentrations of diazoxide, 1, 3, 10, and 100 micromolar. And we measure glucagon secretion in the same islets that we used for the electrophysiological measurements, and these are the responses. You can see that when we increase the KATP channel activity above what you had at 6 millimolar glucose alone, you get stimulation of glucagon secretion, although the increases are very, very small. And then as you increase the KATP channel activity further, you again get a secondary inhibition or suppression of glucagon secretion. This is a more schematic way of demonstrating is you have a bell-shaped relationship between KATP channel activity and glucagon secretion. And what we believe is, and what we observe experimentally, is that in a healthy alpha cell, you sit somewhere here on this bell-shaped relationship. And that means that when you increase the glucose concentration, you produce hyperglycemia. You reduce the KATP channel activity, and then you move to the left of the bell shape. So you get inhibition of glucagon secretion. And when you produce hyperglycemia, you increase KATP channel activity slightly, and that leads to stimulation of glucagon secretion. Now, what we speculate is that in the diabetic alpha cell, for some reason, you have an increase in KATP channel activity. So rather than sitting over here, you sit over here. What happens now when you increase the glucose concentration and you presumably decrease the KATP channel activity? Well, now you move from here up to here, and that leads to stimulation of glucagon secretion rather than inhibition as we observed over here. And conversely, when you lower the glucose concentration, you get the an increase in KATP channel activity, but rather than stimulation of glucagon secretion, we get a further inhibition of glucagon secretion. So this is, of course, based on, this was just a hypothesis, but we then tested this hypothesis using diazoxide. And we used a dose of diazoxide which was very low, and just barely produced any increase in KATP channel activity. And this concentration, it led to the activation of the 3 to 4 channels. And we did this experiment in non-diabetic human pancreatic islets. We exposed the islets first at 1 to 6 millimolar glucose, and these are under control conditions where glucose produces a very good inhibition of glucagon secretion. We then repeated the experiment in the presence of diazoxide, and what you can see is that glucagon secretion at low glucose is much lower than under control conditions, and an increase in glucose now produces stimulation rather than inhibition of glucagon secretion. So this of course echoes perfectly what we see in diabetic islets. And the big question, of course, now is does this really have anything to do with what happens in type 2 diabetic islets? So what we have done here is now that we have used the few diabetic preps that we have. We have measured glucose secretion in response to an increase in glucose from 1 to 6 millimolar. And we express the data here as the percent reduction of glucagon secretion. And as you can see, in non-diabetic islets, glucose produces an inhibition of glucagon secretion by about 40 to 50%. When we repeat the experiments in type 2 diabetic islets, you don't see any inhibition. If anything, glucose tends to stimulate glucagon secretion, but the responses are quite variable. Now what we do is that we see if we can shift this, and this, of course, what we see in the diabetic islet would fit with this concept that we have shifted, or we moved on the bird shape of KATP-channel activity from here to over here. So now we test what happens if we try to normalize KATP-channel activity in the alpha cells. And we use that then a very low concentration of tolbutamide. This dose of tolbutamide that we use is only 10% of that we normally use to stimulate insulin secretion. And if we now use the same islets as we have here, and we measure the effects of glucose in the presence of this low concentration of tolbutamide, you can see that we now restore normal glucose-regulated glucagon secretion. And the reason for that, we would like to think, is that because we have reduced or moved from here to here on this relationship between KATP-channel activity and glucagon secretion. And now, when you increase the glucose concentration or lower the glucose concentration, you produce the normal responses to normal glucagon secretion responses. Why should KATP-channel activity be increased in type 2 diabetes? Well, we don't know, but there is interesting data from studies in skeletal muscle and suggesting that there is a mitochondrial dysfunction both in diabetics patients and also in the elderly, and it's the oxidative phosphorylation that becomes impaired. So we wondered whether this could somehow be related to this age-dependent increase in diabetes incidence in diabetes. So this is diabetes incidence plotted against age, and as you can see, it increases steeply as you go up above 45 or 50 years of age. And I think the idea is, or the popular idea here is that this is due to reduced insulin secretion. But now working on all these human diabetic or all these human pancreatic preparations that we have had, we could then group the islet preps according to age and measure insulin secretion. And much to our surprise, what we observed was that there was no age-dependent reduction of insulin secretion in response to high glucose with age. If anything, there is a rather dramatic increase in insulin secretion with age. Now, what about glucagon secretion? Well, here we see some really interesting differences. So in young people, high glucose is, of course, a very powerful inhibitor of glucagon secretion, and that is true up to the age of 50. And between 50 and 59, the responses are slightly reduced. But even in this age group, every human islet prep responds with a reduced glucagon secretion in response to high glucose. But in the highest age group, between 60 and 69, there is no longer any statistically significant suppression of glucagon secretion. 70% of the preps still respond with suppression, but in 30% of the preps, there is actually a stimulation of glucagon secretion. And this is even in donors with not diagnosed with type 2 diabetes. And to us, we would like to speculate that maybe an increase in glucagon secretion rather than a decrease in insulin secretion might be the primary event in type 2 diabetes. And as to the possible involvement of the mitochondrial dysfunction, this is again speculation, but it is interesting that if we measure glucagon secretion in these non-diabetic old donors, we see this stimulation of glucagon secretion, which is very similar to what we obtain if we use oligomycin to poison the alpha cells and to impair mitochondrial function. But as my colleague pointed out to me before this web seminar, age is, as such, should not be regarded as a poison. But it's interesting that in some donors, it suggests that there is a mitochondrial dysfunction in the alpha cells, and that might explain this regulation. Finally, in the last two slides, I just mention the delta cells, because the delta cells are almost completely ignored in diabetes research. And one of the reasons for that is, of course, that they are very difficult to work with. There are so few of them. I mean, up maximum 10%. But they are extremely interesting. They're extremely beautiful. They have have neuron-like morphology, and they actually connect with many beta cells within the islet. And we have estimated that one delta cell could make contact with up to 20 beta cells. And the reason I want to mention this is because we have recently measured somatostatin secretion from diabetic islets as well. And I've already talked you through the data in diabetic islets on insulin and glucagon secretion. But as you can see here, there are very dramatic disturbances of somatostatin secretion in type 2 diabetic islets. There is a massive oversecretion of somatostatin at low glucose concentration, and there is actually no further stimulation by high glucose. So overall, just looking at this picture, you can see that the effects of diabetes on insulin secretion are actually the least dramatic within the islet of diabetes, and that the effects of type 2 diabetes on glucagon and somatostatin secretion are much more dramatic. And I think it's fair to conclude that diabetes is certainly not just a question of just insulin. Diabetes is certainly hormonal. It might even be trihormonal. And I think the present focus on the beta cells is perhaps not warranted. I think we should devote much more effort to studying the non-beta cells within the pancreatic islet. So this is just a brief reminder of what I've tried to say. And I hope to illustrate that electrophysiology is useful, and that type 2 diabetes involves both impaired beta and alpha cell function, that the alpha cells and beta cells are actually very similar, that the normal regulation involves KATP-channels, and that in both beta and alpha cell diabetes is associated with increased KATP-channel activity, and the secretion defects can be corrected by sulphonylureas. I didn't have time to talk about the GLP-1, but maybe another time. And then finally, we have this exciting data with somatostatin that we are currently working on quite a lot. And this final slide just acknowledges the people that have been kind enough to support our research, and this shows Oxford on a beautiful sunny day. And it actually looks a little bit like that today. So thank you very much.
Thank you. Thank you for a really eloquent presentation of what are undoubtedly very complex pathways.
It is complex.
So we have run over, but I do want to get to a couple of questions if we can. We'll start with this one. Are there no immortalized human islet cell lines available, or are these unsuitable for electrophysiological examination?
There are human beta cell lines, and the Endo-C beta H1 and beta H2. We are working on them. They are actually quite similar to human beta cells. They are not easy to work with, but they are very similar in terms of the ion channel complements and their exocytotic responses. Insulin secretion, their secretory responses in insulin secretion assays are maybe limited to a 2 to 3-fold stimulation, at least in our hands. But other people have been more successful. So there are human beta cell lines, and they are good, and I think they are being improved more and more. Alpha cell lines, we would very much like to have a human alpha cell line, but apparently that's very difficult to establish.
Okay, and the next question. Based on what you presented, the timing of when to collect samples for insulin and glucagon testing is absolutely critical. What guidance could you provide when assessing hormone results to enable to detect clinically significant responses?
Well, I mean, I think it's the ideal situation would be to take as many samples as you can, and that the patients permit. I mean, when we work on rodents, of course, we are limited by the small blood volume, so we can't take that many samples. But in people, you could take many more. And the response to glucagon, I mean, alpha cells respond very quickly. to glucose. So, I mean, within 5 to 10 minutes, you should certainly see a suppression of glucagon secretion, and that would coincide with first phase insulin secretion. So if you only want to take 3 samples, that would be a baseline value, maybe at 10 minutes, and then maybe at 60 minutes after a glucose challenge or a meal tolerance test.
Okay. Does bariatric surgery improve the first phase of insulin secretion in type 2 diabetic patients? And if so, what is the mechanism if their beta cells have intrinsic defects as you described?
Yes, I believe that bariatric surgery improves first phase insulin secretion. It certainly improves insulin secretion in diabetic individuals, and we have done a little bit of work on this that we recently published. That in diabetic rats, bariatric surgery leads to a dramatic improvement of insulin secretion and beta cell function, but it also improves and corrects alpha cell dysfunction. And we think, but this is maybe a bit controversial, but we think that this is mediated by PYY, because if we take islets from diabetic rats and treat them with PYY, we have the same effects in vitro. And if we take serum from rats that have undergone bariatric surgery and treat human islets or rat islets with, if you get rat islets from diabetic rats with serum from rats that underwent bariatric surgery, we can reproduce that effect, and the effects can be removed if we immunoneutralize PYY. And PYY also has a very good effect on the performance of human pancreatic islets. So I think, what I would guess is that bariatric surgery, possibly via PYY, it leads to a normalization, or it repairs the dysfunctional beta cell, and we see actually changes in gene expression in the islets following bariatric surgery and following a PYY treatment.
Okay, the next question is, are there any working imaging-based techniques to monitor the membrane potential of alpha and beta cells with sufficient resolution?
If the question is whether this is in vivo, the answer is no. If the question is whether if there are such techniques in vitro, the answer is yes. These techniques are. We have been trying to establish and working on them in isolated islets. It requires some rather sophisticated hardware to perform these measurements. But we have occasionally seen a few action potential like things. But this is certainly something which we would like to do. It would be much nicer to be able to record changes in electrical activity using light rather than using electrodes. It's less invasive, and of course it's probably less difficult.
Have you measured the ATP-ADP ratio in alpha and delta cells from diabetic patients?
No, we have not, but this is something which we would very much like to do. There are ways of doing this online using fluorescent probes, and so we would, when I was preparing this web seminar, it actually occurred to me that we have now the methodology to make such measurements. So the next time we get a diabetic prep, which I don't know when will happen, I will certainly persuade one of my team to do such measurements.
Okay, and the last couple of questions. In individuals with metabolic syndrome or prediabetes, please provide your guidance on the suitability of using both insulin and glucagon testing to monitor progression towards or regression from type 2 diabetes.
Insulin is, I would say, the best I think one of the best tests of beta cell function, I think, is arginine. So arginine is something which is an amino acid, and it stimulates both insulin and glucagon secretion in the islets. And it does that in a way which is fairly physiological, and you can use that to estimate. I mean, the responses, I think, would provide an indirect measure of the functional capacity of the alpha and the beta cells. So glucagon tests, I think, would be. I'm not familiar with the glucagon test, but I think arginine would be a better test. I mean, just to test the remaining beta cell capacity and its progression. And it would, and if you complement that with a glucose test, I mean, it would actually allow you can see how the responsiveness to glucose and arginine changes with time, and that would be a way of monitoring whether it's a reduction of the number of beta cells or whether it's just the responsiveness to glucose that is affected. Because arginine would bypass; it would act in a torbutamol or sulphonylurea-like way on the beta cells.
And finally, are there any differences in glucose-mediated responses in freshly isolated islets versus islets that have been kept in culture for days? Yes.
We see that, the effect is particularly pronounced for glucagon secretion, we find. Insulin secretion, that's fine. But alpha cells, we see just over 24 hours of culture, we see that basal glucagon secretion is reduced by 75 to 80% compared to freshly isolated islets. And then, of course, you could argue that maybe the reason that the freshly isolated islets are hyper-secreting is because they are slightly damaged. But I don't think so. Because, if you try to work out the release of glucagon per islet and compare that to what we observe in the perfused pancreas, where there's been very little manipulation of the islets, then the values that we get in the freshly isolated islets closely correspond to what we see in the perfused pancreas, whereas those that we observe the next day of 24 hours in culture are too low. And exactly. So I think that maybe perhaps a factor that contributes to the lack of consensus when it comes to the understanding of the mechanisms regulating glucose secretion is that many labs, and we have done so in the past as well, culture the islets before they start working on the try to measure glucagon secretion. And maybe there are changes due to the culture of our alpha cell function, which have a confounding impact on the experiment. And at least one should keep data obtained with cultured islets and freshly isolated islets separate. And I'm not sure that distinction is always made. Okay.
Okay. Well, I'd like to thank our distinguished speaker, Dr. Patrik Rorsman, and thank you to everyone who joined us. We hope you enjoyed this webinar and also hope you have a great rest of your day.
Thank you.